And today, like though the whole motivation is to uh, give an insight about the artificial intelligence, sadly, I am going to talk about the importance of having rational design in the age of artificial intelligence. And with one example, I will try to show that how we still need to generate data so that we can train different models to like generate different kind of folds or different kind of structures. So before I get into the uh, field, let me say a few words about my lab. I am a new joinee in IIT Palakkad, joined last November or October end, uh, and uh, I will be working on protein design. As Asha said, this is like IIT Palakkad, which is still evolving and promising to be a vibrant place. We are developing IIT. What I will be doing is the computational design and and validating them in the lab for a different functions. If we define that, like I will be using different databases, web servers, designing different databases and web servers, training the models to design proteins, and then that proteins will be with the focus on some function, and we will validate them in the wet lab, and the learnings will be taken back to the model, and we will refine it, optimize it. And this is the process I am planning to continue in the near future. To give you one example, the basic focus will be on the thermophilic proteins. For example, what we can do, we can see is this is the complex one, which is like a mega Dalton protein, which transports electron from one place to another inside the, like this is the part. So what I'm saying, like this may not be needed, this full structure may not be needed because the proteins are evolved for their function, not for their structure. Can we design a minimal structure, let's say four helix bundle, which has got all the uh, electron hubs in the terms of pairs and sulfur uh, uh, sites, so that electrons can jump from one place to another. If we can do that, then can we just extrapolate it by end-to-end stacking of these proteins or these four helix bundles to form a wire? If these things are done, that will be cool. But this is one example why thermophilic proteins I am focusing on, because many of these functions will be carried out at a higher temperature. So this is one of the example where we'll be using the computational biology approach to design proteins, AIML to design these, and then the experimental to validate. And wherever I am not able to, then collaboration is the next goal. So having said that, like why, how, and just to prove like okay, I can do all these things, let me take you back to what I have done in my past. During my PhD or during my postdoc, I developed many algorithms. These algorithms are really used by the scientific community, but as a developer I developed these algorithms, but as a user also I use these algorithms to design or to analyze different secondary structures, and then to analyze the oligomerization or homomerization of different proteins which are generated by AlphaFold, and we were successfully able to predict the oligomeric state of different proteins, and that was also very useful for their sequence to structure to function relationship. But as an experimental biologist, I I designed the first ever 310 helical bundle, and then that was completely rational design approach. But using machine learning approach, we were able to generate the seven helix coiled parallel single chain protein. But today I'll be focusing more on the design of 310 helical bundle. And I must say, I did this work when I was part of Jack Wilson group at University of Bristol as a postdoc assistant. Then I have talked so much about these proteins. Then what is protein design? So the point here is you have a sequence, you can find whether that protein can fold to a given structure. Nowadays, there are like metamorphic proteins. You can find lots of structures for a single protein sequence. But when there is a sequence to structure, you say it protein folding. And when you have a structure known, which sequence can adopt that protein structure, this is called protein design. To just give you a rationale, what happens like when you go to the clothes shop, and a shopkeeper says like, sir or madam, like this cloth will really suits you. Actually that person is a protein designer acting as a protein designer, where you are seeing you and then trying to suggest a better suit or better clothes for you. But what is the idea behind it? The idea of this protein design field is, how can we make the protein easier, predictable, and more reliable? The idea is, like, if I give you a pen and paper and ask you to write a sequence for a particular fold, and if you're able to write that, I will say like you understand that fold very much. And that's the protein engineering, protein design approach for that getting the sequence. And the basic mantra behind it, what I cannot create, I do not understand. That I am not saying that Richard Feynman said when, like, it was written on his blackboard. Mm. And this drives the field, or I think this drives any field for that matter. So now that we know what is protein design, but let me explain what protein structure is. Mm. So this is what I feel about the protein structures. Given that you have a protein structure, it is nothing but all the helices joined to each other by loops in general. Like you have several nowadays, several proteins which with no secondary structures, but those are like very few. For time being, let's say these protein structures are defined as a helix plus loops. In this current talk, I am going to talk about alpha helix inspired design of a 310 helical 310 helix. But the point is like why I chose 310 helix, and the answer lies in the timeline. You'll see like this 310 helix term was there in since 1941, and 1941 is like there is a year came like 1951 when when Linus Pauling proposed the model of alpha helix. So you can see like this 310 helical term is there before alpha helix was coined. But over the year, what you can see, like these 310 helices are actually remained at a very basal level of research, or like 1953 it was proposed as a like a single helix, what will be the hydrogen bonding pattern, then people started how we can make it, and then they remained there as a single helix. How can you make these single helix 310 helices? Contrary to that, alpha helix, proposed in 1951, 1953, Crick model came for the coil, 1972 to 2000, then lots and lots of softwares and databases to find these alpha helical coils or different alpha helical assemblies, and 2000 onwards. So before that also people started associating these functions to the alpha helical assemblies. So what we can see is that 310 helix field was really lagging and still at the stage of like a single 310 helical term. But the question is, why do we need to have 310 helix or their assembly? And the question also comes, when the nature doesn't want 310 helices, why we are going behind these 310 helix? So idea is also that like what these 310 helices, like as a protein designer, can you explore the dark matter? Whatever is not there, can you show your creativity and find something that is not there and make it functional, or make get some function from that assembly or whatever you have made, but in a better way, in an efficient way. So it's not like 310 helices are not present in the nature. They are there, very short, and they are also involved in really either structural properties, or they have sometimes associated with functional properties also. But why I was interested in 310 helix was because of the structural diversity it was going to bring to my plate. Is like I now have another brick which is 310 helix. Using that, I can now design different modules of the protein structure, and then I can use them to design or to have those complex protein structures and get them functional. Like and if I can find the conversion interconversion of threaten and alpha or alpha pi or something like that, then it will be really easy for me to have a dynamic function associated uh, associated with these structures. So now let's come to the Cretan helical uh, field again. Uh, and I'm sorry, like this will be more like a boasting for this work, where I will be showing like how many challenges there were, and still we wanted to go ahead with the Cretan helical quaternary helix design, quaternary structure design. So in nature, Cretan helices are really less abundant. In fact, only 4% of the residues are Cretan helix. And that also, if you go carefully, then you will remove those helices really significantly, because like all of them are really short, 3 residues, 4 residues long. So in fact, only like around 1,000 of helices, Cretan helices are more than 6 residues long. And why is that case? Because if you see the energy contour on the Ramachandran plot, what you can see is like this blue star is for phi psi for alpha helix, and red star is for uh, 310 helix. What we can see here, like any peptide you design to be in 310 helix, the energy well for the alpha helix is so steep that it will fall into that trap and we will never get a 310 helix. It was then really necessary to find an additional stabilizing factors for these 310 helixes. And people have tried this. And what we can see here, I am is hung. Okay. What people have tried is like use of different quaternary amino acids to induce 310 helicity in a designed peptide. Our obvious choice became AIB or alpha isobutyric acid. Why this? Because this is the simplest among these three, at least what I am showing. There are many, but believe me, these derivatives are also quaternary amino acids. And the point is like having quaternary amino acids induces some stress on the C alpha, so that the angle between NC alpha and C is no more 109 or 109 point something. It became around 106 or 105, so that like there is a bend, a higher bend, and then the 310 helicity is more preferred. But when you do the propensity check in the or the percentage presence check of AIB in different peptides or different proteins in PDB, what you can see is like all those AIBs, majority of them are in alpha helix, very few are in 310. Also, like though I'm showing this, but there is a catch. These AIBs are not used like whatever they're in the alpha helix, they are single or double, or like one or two AIBs are present in tandem. Hence, there is no induced effect of AIB on the helicity. Hence, they are mostly in the alpha helix. But wherever they are they are present like a 10 or 11 residues or 8 or 9 residues. They are in 310, and these are shown here in as like 33 in number. So this was a challenge because the number was not like suggesting us to go with the AIB, but we were also excited with the fact that this is the simplest one, and it can lead to the 310 helicity, whereas the normal residue like non or standard amino acids like 20 standard amino acids, we are not pushing any peptide towards the 310 helicity. But again came there was a challenge, was a Carle Balram plot. Carly Palram plot suggests like if you have any peptide of length say n and number of AIDs in that peptide, let's say m or x, if the peptide length n crosses 8 or 10 residues, no matter what's the value of x, you will never get the 310 helicity. That will be invariably alpha helix. In that case, we were also having a challenge of how much the length of the peptide going to be, because we were going towards the quaternary structures of problem, and hence we wanted those peptides to come together. Then the question was, what is the length of these peptides going to be? So my approach towards building these 310 helices was actually getting from alpha helix, coiled coil. This is type N coiled coil heptad repeat, which is, you can see that A, B, C, D, E, A and D positions are hydrophobic, and delete the F position that I'll explain in subsequent slides to get the 310 helicity. Like if delete the F position and hope that there will be assembly. Why? Because only 310 in this case, when you have deleted the F, only in 310 helical conformation you will have the assembly, because then only these hydrophobic residues will be pointing towards one side. We ask these questions. What is the nature of the sequence of repeat going to be? What is the length of that peptide going to be? Which factors, like what are the stabilizing factors, and how these helices are going to come together? Let's come to the coiled coils then. What coiled coils does is like it has got type N to C, we are seeing from top to bottom, and then A and D are actually hydrophobic residues, G and E are polar. F is like you can add anything over there, any helix inducing residues. B and C generally alanine, but you are free to have anything. It is a type N, otherwise B and C will also change. So now what happens? Because A and D are in line, then there is a hydrophobic seal and there is a polar part. It is empipathic. In order to avoid water, these A and D, because of these A and D hydrophobic residues, these helices come together like this, and they form a coiled coil. A typical coiled coil has a knob into hole interaction, which is shown here. But here was a problem. So you can see like this is the 310 helical conformation I was similarly I was expecting. Like if we can have a head of hydrophobic vertex and then the polar vertex, then it can be empipathic nature, and then this can come together and it can form like a 310 helical assembly. But the catch for the alpha helical coil was like you change A and D residue and you will change the uh, oligomeric state. Like here at A position, you have I, and D position L, this is dimer. You have Li, it is a tetramer, just reversal of A and D. Like everything remains same. You have to just see. And if you go to the PII, like all the A and D positions are isoleucine, it will become trimer. So the choice of hydrophobic residue was also important. But you can also ask at this point, why do you even worry about these hydrophobic residues? Because you don't know what's the structure going to be for the oligomeric state, or for that matter, you didn't know whether it is going to form any 310 helix or not. Because we were really starting from the scratch, and we really starting from the really less number of next to no data available. But anyway, so like these answering these first three questions: what is the nature of sequence? What is the length of peptide, and which factors are going to bring these helices together? We turned to the bioinformatics, and we then went to all the PDB files available, and we found that all these are the criteria that we applied. 22,000 310 helices we got, 67,000 alpha helices we got from the 10,000 protein structures. Now, this is really huge number from the 310 helices, where I was saying like only 4%. So actually, these 310 helices are like, if you see, So I'll come to like what is the actual number, but the idea here was like at least to prove that geometrically they are different than the alpha helix. There is a difference. Like these are two distinct entities. What we did, we just plotted their length variation, their rise per residue, radius and residues per turn. We were really focusing on rise per residue, radius and residues per turn because the residues per turn was going to define the repeat length of the repeat. Like for coiled coil it is 7 heptad repeat we call. So what like that, and heptad repeat gives you like 2 turns of the alpha helix. And here the 310 also we wanted to know what is the number of residues per turn so that we can take some integer value very close to that. So we found that for 310 helices, like this chart is showing you these 310 helices are really different than the alpha helices. Then this 310 helix has 3.1 residues per turn. So we were happy with the 6 residues per repeat, 6 residues repeat. And radius is also significantly different. Like what we are showing here is like 310 helices are really thinner or thinner than the alpha helix. Uh, but what you can also see here, the length distribution, number of residues per number of residues for 310 helices. Out of 22,000 310 helices, these are actually like 17,000, almost 17,000 helices are 3 residues long. So you really don't know what these helices are, because for any helix to be defined, you need actually two hydrogen consecutive two hydrogen bonds to be called as a or called as that type of helix. But anyway, so now we knew that the repeat is going to be 6 residues repeat, or I will call it now hexad repeat. But which residues to choose? And mainly we were focusing on hydrophobic residues. Why hydrophobic residues? Because like I showed you like these choice of hydrophobic residues was really important because the oligomeric states change. But I will not show you that data. But later we figured out, uh, apart from leucine, anything you choose that is not going that peptide didn't fold to any 310 helix or for that matter alpha. But what we found here is like all these three hydrophobic residues, uh, when we divided it into like database was divided into two parts. One is the longer 310 helix and one is shorter 310 helix. Longer 310 helix like with with 2 turns or more, and the shorter is less than 6 residues or 2 turns. Longer helices are shown in the orange color, and for leucine, what you can see is they are really preferred in the longer 310 helices. And we had the first choice for the hydrophobic residues, and the two positions were really fixed, and it was leucine. Then the obvious question was like, what is the length going to be? From our coiled coil design part, we knew that if we have 4 repeats of heptad, then they have enough hydrophobic seal so that they can bring these helices together. Then we said like, okay, if that is going to be enough, then we also will go by the 4 residues, four hexad repeat. Later we did for the 3 repeats and 2 repeats also, and we also went to the 5 repeats. And the idea was at what length the 310 helix is actually stable enough from at the lower concentration of the peptide. So then when we had all these residues fixed, and then if you see the alpha helical coil assembly, and this is the heptad repeat, what the repeat goes like E L A L K Q. Very simple design. We removed Q. These alanines are replaced by AIBs. E and K remained the same. Choice of L came from bioinformatics analysis. So this was the design principle, and then we started we made a series of peptides starting from the alpha helical coiled coil ELAALKQ repeat. and obviously that was going to be the primer, and it remained primer. And when we replaced alanine by AIB, it still remained dimer, coiled coil. And the conclusion from this is like, you know, AIB on its own is not sufficient to have a 310 to induce a 310 helicity in a longer peptide, very much showing the same thing what Carl Balram plot was has shown long time back. But then we turned to hexad repeat, and we again started from this timer, and we removed this F position. The sequence is here. It again remained alpha. It again remained alpha helical, but this time it was monomeric, only 30 to 40% helicity. But the magic happened when we replaced this alanine by AIB, and it went to the 310 helicity. Here you can see the 310 helical assembly that we got. And yeah, so the point what I am trying to make here is like AIB on its own cannot induce 310 helicity. It requires the interaction of these helices to come together along with the presence of AIV, and that will induce the 310 helicity in the peptides, and you can have the 310 helical uh, oligomer. But just have uh, a bit deeper look into these 310 helical assembly is something like what you can see here is like this is octamer that we got. It is nothing but two squares with same center with some angle, like one inner square, outer square. Inner squares are making the hole, and the outer square is making a knob, and there is a pseudo knob into hole interaction and stabilizing the full helix assembly. So then this is the octamer. From the study also uh, we saw that like this is octamer from the biophysical data we were having. Like, we knew that it is going to be higher oligomeric state, and we knew that okay, the main aim to design such a large assembly is to get it functional, and the basic function that we can install is something like what we can insert into the whether we can use the core or not. So first foremost, what we wanted to know, like whether there is any channel in the assembly or not. From our alpha helical coil, we knew that the like the higher oligomeric coil have a channel inside. But sadly, for these 310 helices, they were not binding to DPH or prodan, and these are like they bind to the hydrophobic core and gives a signal, fluorescent signal, so that like you can find like where there is a hydrophobic core or not. So the binding was not very specific. So what we thought then, like why don't we engineer it inside uh, that assembly? And for that purpose, we replaced these leucines inside by the alanine. And why we replaced two leucines to mutated two leucines to alanine? Because the length of this protein is actually very similar to the length or the core size we were getting here. And then we did the again binding assay with the prodan, and this time it was a saturated binding, suggesting there is a specific binding of prodan to the hydrophobic core. Then we turned out to like by then like what happened there was AlphaFold came into the picture. Like people were going behind AlphaFold. Oh, AlphaFold has solved everything. We thought like okay, fine, now AlphaFold has solved everything, let us check all these structures whether we can generate with the AI or not. So first thing first, like we had the structure. We then said like okay, given the structure, whether we can predict the sequence or not. And there is a software called Protein MPNN. So we said like, okay, protein MPNN, this is the structure, give us the sequence for these this structure. And they gave like lots of structure, lots of sequences. And we said like, fine, let's go and check. So first question was like whether these sequences are actually like are like whether AlphaFold can predict these sequences uh, to be 310 or not, and the answer was no. It was not able to predict it as alpha helix as a 310 helical assembly. Actually, it was predicting it as a alpha helical assembly. Uh, but the confidence was really less. Nevertheless, we were happy with this confidence score that AlphaFold is, though it is not predicting the structure to 310, but the confidence is low. So like, let's check in the lab. Let's go to the lab and check. So I took the first two sequences, or first 2 or 3 sequences. I'm showing data for the first sequence, and went to the lab, made the synthesized that peptide, and the CD is something. It shows that like very few, very little helicity, and it is actually an aggregate in solution. So having, like, why I'm showing you this data, because like with this, what we were trying to show, like though the AI was able to solve lots of protein structures, and like it's great to have, but all these like less frequently occurring structures where people are not working or not enough data is there. So like we need somebody to generate that data so that AI can get uh, trained on, and then later can use can start predicting more robust sequences or structures for those type of secondary structures or folds. So the take-home message from this is we were successfully able to form a 310 helical assembly, which is like a really longest by far. Like the longest what was available was 13 residues, and this is like 30 residues long 310 helix, so longest by far. We were also, I did talk about, but it is available in the paper, the longest, the hetero system, where two type of different helices can come together and still form AB system, and in a one-to-one manner. Then we also showed a point where the leucines and AIB are can induce the changes in the helicity from alpha to 310. This was the length dependent dependence I was talking about. We went to 4, 3, 2, and the two hexad repeat was actually alpha helical, and the 3 residues was really cool helix. Like it was alpha helix at lower concentration, but then it changed to 310 helix at the higher concentration. So it is the concentration induced structural change into the peptide. Mm, but the bottom line is AlphaFold and other related algorithms are really great. They are doing a great service to the community, but non-standard structures are really a challenge for these AI tools, and still we need to work rationally to achieve that structure. With this, this is my thank you slide. It's actually a thank you peptide, or OA